Hello everyone, welcome to Genetic Concealed. I am your host for the Women Alessio Deleke, and today I'll be covering 3 topics, 2 articles and 1... okay. RNA-based medicines. RNA medicines are one of the biggest breakthroughs in modern science, especially after the success of the COVID-19 vaccines. These treatments can target disease by shutting down specific genes, but there's a problem. How do we get these molecules into our cells effectively? A new study published in eLife on February 4th might have some answers. Researchers at the University of Maryland studied tiny roundworms to understand how double-stranded RNA enters cells. They found multiple ways these RNA molecules move between cells and even across generations. One key protein in this process is called SID1, which helps transport RNA. Surprisingly, when researchers removed SID1, the worms became better at passing on gene expression changes to their offspring. This suggests that RNA might have a much bigger role in inheritance than we thought. And since humans have similar proteins, this discovery could help us develop new treatments for inherited diseases. Another interesting find was a gene called SDG1, which helps control jumping genes, which are pieces of DNA that can move around within the genome. While jumping genes can create useful genetic diversity, they can also create harmful mutations. The fact that SDG1 regulates this movement could lead to future genetic therapies. So what's the takeaway? Scientists are still uncovering how RNA works, but these discoveries could lead to better treatments for genetic diseases and even ways to influence inheritance itself. RNA medicine is most definitely an area to watch. Now, if you're studying biology, you've probably come across this. But at the very least, I have. Epistasis. Epistasis occurs when one gene affects how another is expressed. This means it's not just about dominant and recessive alleles. Sometimes a gene at another location can completely mask the effect of another gene at a different location. There are 2 types. Okay. Recessive epistasis. For example, in salvia, there are 2 gene loci involved. Upper A, lower A, upper B, lower B. The upper B allele makes purple flowers, while lower B makes pink. But here's the twist: if a plant is homozygous recessive, the flowers will be white, no matter what's at the B locus. This means homozygous recessive epistatic to B. It stops B, upper or lower B, from having any effect unless at least one dominant A is present. Now, how is epistasis different from codominance? Another term is codominance, where both alleles in a heterozygote are fully expressed. Examples include human blood groups. If you inherit A and B alleles, you get A blood type expressed in both proteins. Catcoat color. A red and white allele together gives a speckled growing coat. Epistasis is super important in genetics because it helps explain why some traits don't follow simple Mendelian ratios. If you're doing A-level biology, make sure you understand this, as it could come up in exams. Now, for the second article. Have you ever wondered how likely it is that aliens exist? A new study published in Science Advances on February 14th challenges an old idea that intelligent life is extremely rare. For years, scientists followed something called the Hard-Steps model, which argues that intelligent life is incredibly unlikely because it took so long for humans to evolve. But new research from Penn State suggests the opposite, that intelligent life could be a natural result of planetary conditions improving over time. Now, what's so different about this new model? Instead of assuming life is just about luck, this model suggests that as planets change, life adapts. On Earth, oxygen levels increase over time, making it possible for complex organisms to evolve. The researchers argue that humans didn't evolve early or late, we evolved when conditions became right. And what does this mean for finding aliens? If life follows environmental changes, then intelligent civilizations might be more common than we thought. Scientists are now focusing on searching for biosignatures like oxygen in exoplanet atmospheres, studying how microbial life evolved under different planetary conditions, re-examining the idea that life requires lucky breaks, maybe just about the right conditions appearing. This is an exciting shift in thinking. Instead of seeing human intelligence as a one in a trillion event, there's more to suggest that life, maybe even intelligent life, could be waiting for us to find it. Maybe we're not alone after all. No matter, even with all this, I still believe in the superiority of humans. May God save us. That's it for today. Never forget, genetics is beautiful. I'm your host, Follow me, Alessio Deleke, signing out.